Hello students, today we are starting with biology class 11th chapter number 7 cell division. Okay, we have already started this chapter in our last lectures. Few lectures have been done with this chapter. Okay, we have studied some important topics, that is we have studied about mitosis, cell cycle, then we have studied the stages of mitosis, that is we have studied about the prophase, metaphase, anaphase, telophase. We have studied about the cytokinesis in plant cell, animal cell, significance of mitosis. Okay, and then we have started with meiosis in our last lecture. So when I say meiosis, let us revise the definition of meiosis. Okay, we have started with the introduction of meiosis, but just we will revise the definition and then we will continue with the further topic. That is, meiosis means the form of cell division by which gamete with half the number of chromosomes are produced. Okay, so meiosis is a process in which half the number of chromosomes are produced. We have studied in detail about this in our last lecture. So let us move forward. That is phases of meiosis 1. Okay, there are two types of meiotic division, that is meiosis 1 and meiosis 2. So we have started with meiosis 1. There are sub stages in meiosis 1, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. When I say the subphases, we will study each subphase in detail. When I say prophase 1 consists of substages, that is leptotene, zygotene, pachytene, diplotene and diakinesis. Before studying all these substages in detail, we need to study some terms that are going to come in this prophase 1. So let us start with some of the terms that will be involved in prophase 1. Prophase 1 is the longest and most complex phase. That is 90% of the time the cell is going to be remain in prophase 1. Chromosomes at this phase are condensed. Synapses occurs during this phase. That is homologous chromosomes come together to form tetrad. Tetrad is 2 chromosome or 4 chromatids, that is sister and non-sister chromatids. So you have came across many words in this slide, that is synapses, homologous chromosome, tetrad, chromatids, sister and non-sister chromatids. So now this, before studying in detail the substages of prophase 1, we will first study the terms about the terms that are going to come in this phase. That is synapses, homologous chromosome, tetrad formation and sister and non-sister chromatids with the help of the diagram. So see here, this is the synapses. You can see here homologous chromosome. This is homologous chromosomes. These are homologous chromosomes. These 2 chromatids are known as sister chromatids and these 4 together they are known as tetrad. Okay, now one by one we will study in detail about these terms. What is synapses? What is sister chromatid? What is tetrad? Okay. Let us first study about the homologous chromosome. Homologous chromosome means what? The pair of chromosomes, that is maternal and paternal, means one will be maternal chromosome, one will be paternal chromosome. The pair of chromosomes that are similar in shape and size are known as homologous chromosome. The pair of chromosomes, maternal and paternal, that are similar in shape and size are known as homologous chromosome. Homologous pairs, tetrads, carry genes controlling the same inherited triads. Okay, homologous pairs means tetrad. You can see here, these are homologous pairs. They are forming the tetrad. 4 chromatids together, they are known as tetrad. So, homologous pairs, tetrads, carry genes controlling the same inherited triads. Each locus, that is position of a gene, is in the same position on homologs. Okay. Humans have 23 pairs of chromosomes. Human beings are having 23 pairs of homologous chromosomes. Out of that 23 pairs, 22 pairs are autosomes and 1 pair is of sex chromosome. So, 22 pairs are autosomes. This autosomes means what now? Autosomes means the pairs of chromosomes that are responsible for the other body characters other than the sex chromosomes. Okay, so the autosomes are the chromosomes that are responsible for the body characters other than sex-linked triads, that is sex chromosomes. Okay, and the last one pair is sex chromosome. That means in male it will be XY chromosome and in female it will be XX chromosome. So that is the last pair of chromosome that will determine the sex of a person, that is male or female. Next you can see here homologous chromosomes. Okay, paternal homologous chromosome and maternal homologous chromosome. Next term that you need to study is crossing over. Crossing over means it may occur between the non-sister chromatids at the chiasmata. Now what is chiasmata? Chiasmata means they are the sites of crossing over. Wherever you will see the crossing over is taking place, that crossing over region is known as chiasmata. Crossing over means the segments of non-sister chromatids break and reattach to the other chromatid. Okay, crossing over is something that means exchange, interchange of genetic material is going to take place in crossing over. And wherever you will see crossing over, the crossing over region when they are attached to each other, that is known as chiasmata or chiasma. Okay, so let us see this. You can see here sister chromatids. I will explain you when this is known as centromere. Okay, this is centromere. These are chromatids. When two chromatids are attached to a single centromere together, such type of chromatids are known as sister chromatids. Okay, and when the chromatids are of different chromosome, if this is one chromatid and this is another chromatid, then they will be called as non-sister chromatids. But you can see here when two chromatids are joined together with a single centromere, then this type of chromatids are known as sister chromatids. Okay, so sister chromatids, then four chromatids together are known as tetrad, and when the chromatids are same with their size, shape, structure and morphologically, genetically same chromosomes are known as homologous chromosomes. Okay, so all the terms are clear. Chiasmata means what? The crossing over. When the crossing over is going to take place between this tetrad, exchange of genetic material will take place and the crossing over region is known as chiasma or chiasmata. Okay. So today we have studied about the homologous chromosome, synapses, chiasmata, 23 pairs of chromosomes. Okay, so there are crossing over. So there are some important definition that you need to remember before studying in detail the substages of meiosis, that is leptotene, zygotene, pachytene, diplotene and dikinesis. We will study all the substages in detail, that is about leptotene, what is going to happen in leptotene, what is the condition of chromosomes, but there will be all these terms will be involved there, that is synapses, okay, chromosome, homologous chromosome, tetrad, bivalents. Tetrads are also known as bivalents. Okay students, tetrads are also known as bivalents. Remember this. So during that time I will not explain this in detail. So you need to study this before studying the substages. Okay, and homologous chromosome, sister chromatids, all these terms I will be using in further lectures. Okay, so you need to remember all this. So there are some important definitions. Let us go through those definitions and you need to by heart these definitions. Okay, meiosis. First one is meiosis. Meiosis is a process of reduction division in which the number of chromosomes per cell is cut in half through the separation of homologous chromosome in a diploid cell. There is one more definition that we have studied at the beginning. So you can study that definition. That will be easy for you to study. Okay, that is meiosis means what? It is means the division in which the number of chromosomes are reduced to half. Okay, that is the simplest definition. And this definition also you can study, that it is a process of reduction division. I have told you that meiosis is also known as reductional division, because in this chromosomes numbers are getting reduced. They are getting reduced to half. Okay, so we can also say it as a reduction division. Next definition is tetrad. Tetrad means structure containing 4 chromatids that forms during meiosis. I have shown you the diagram of all this, that the 4 chromatids are present together, and that is known as tetrad. Then crossing over. What is crossing over? Crossing over is a process in which homologous chromosomes exchange portion of their chromatids during meiosis. Okay, this also I have discussed. Crossing over means what? The process in which homologous chromosomes are interchanging their genetic material, that is the portion of their chromatids during meiosis. Okay, so in our next lecture we will start with the substages first, substages of prophase 1, that is leptotene, zygotene, pachytene, diplotene, diakinases. And then we will study about the different phases, that is prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinases 1. So we will stop here for today. We will continue this chapter with the substages in our next lecture. Thank you for today. We will continue in our next lecture.